Okay, so good afternoon, everyone. Today I'm going to present the project in Uppsala University, which is investigating isobutene accumulation from Synechocystis PCC 6803 with genomically integrated RNKD, which stands for α-ketoisocaproate dioxygenase from Rattus norvegicus. Okay. So, I didn't prepare an agenda, so we'll just go right into an introduction. Okay. So, as we are all too familiar, the carbon dioxide in the atmosphere continues to increase. And for the first time in human history, atmospheric CO2 concentration reached 420 ppm in May this year. And if this trend continues, the greenhouse effect is going to get worse, which could trigger more severe weather events such as typhoons, floods, and droughts. And since the carbon emissions mostly come from burning fossil fuels, what better way to solve the problem by directly convert the emitted CO2 into fuels, which bring us to biofuels from cyanobacteria. So cyanobacteria are a group of photosynthetic bacteria with their high efficiency in photosynthesis. They have the potential to be a cell factory to convert sunlight, water, nutrients, and of course carbon dioxide into various types of fuels. And in this study, we used Synechocystis PCC 6803 to be our little cell factories, since they are one of the model organisms for cyanobacteria. So among all the possible compounds, we chose isobutene to be the focus of this study. So isobutene is a volatile compound that can be converted into rubber and gasoline. And the reason we chose isobutene as our focus is that it has a large market size, and two, it is volatile, so it's mostly released into the headspace, which will minimize the potential product toxicity. And in the previous study, it was reported that Synechocystis can produce isobutene by introducing RNKD, which comes from the common lab rat. And the isobutene biosynthesis starts with pyruvate and branches off from the leucine biosynthesis pathway. Okay, so how do we introduce the heterologous gene into Synechocystis? So one method is through a replicative vector such as PEEK2. Which is similar to how recombinant proteins are expressed in E. coli. So PEEK2 was used to introduce RNKD in the previous study by Hannah at Uppsala University. However, it is reported that after a few weeks of cultivation, the isobutene production decreased, which is the result of genetic instability. And in this study, we wanted to avoid the instability by integrating RNKD into the genome of Synechocystis with the integrative vector. So how does a gene jump from a plasmid into the genome? So first, we need to pick an integration site on the genome, and then we amplify the homology arm. Homology arms are the sequence upstream and downstream of the integration site. And then we insert the homology arm into the integrate vector, flanking the gene we wanted to introduce. And finally, when you add the plasmid into a Synechocystis culture, the cells will take up the plasmid, and through double recombination, the genes get inserted into the integration sites. So here are the integration sites we chose for this study. So neutral site 1 is a commonly used integration site at Uppsala University, and DDH are commonly used for metabolic engineering to knock out the competing pathway for pyruvate-derived products. And DNAK1 and ARSB are two newly discovered integration sites last year. And in this study, we chose these 4 to investigate the impact of isobutene production when we introduce the KTH into these 4 integration sites. So in this study, I had them all integrated with RNKD, and originally I wanted to introduce another copy of RNKD through a replicative vector, but due to time constraint, I didn't manage to create a proper replicative vector. And due to time constraint, again, only the NS1 KD strain, the performance of that strain was investigated. Okay, so now it's time to investigate the cell's ability to accumulate isobutene in the headspace. So aside from the NS1 KD strain I prepared, I also used the peak-2 KD that's used in the Mostilla study. And so for these 2 KD plus strains, I also prepared their corresponding KD minus strain, which is the peak-2 empty vector control and the NS1 knockout strain. And here are the genotypes. So you can see that all the KD genes are driven by the promoter TLC core and the ribosomal binding site BCD2, except the NS1 knockout strain. And the reason for that is that, once again, time constraints, I didn't manage to create a suitable KD minus control strain. And for each of the KD integrated strain, I also prepared 3 different transformers to investigate if there's any difference in isobutene production among all the transformers. Okay, so for this study, I did 4 experiments. Since I only have 30 minutes to present, I'm only going to present 3 of them. Okay, so the procedure for the second experiment went like this. In this experiment, for each strain or transformant, I inoculated the seed culture from the glycerol stock. And after growing for a week, I transferred them into pre-culture, then growing for another 7 days. And after 7 days, I made 3 sealed cultures in a gas-tight vial. And this 3 is access triplicates. Okay, so in this experiment, I decided to measure the isobutene accumulation at 2 time points to see how the isobutene production evolves. So after the cultures are sealed, I incubate them for 5 hours and then measure the isobutene through gas chromatography mass spectrometry. And then I take the samples for all the measurements. And then I incubate them for another 12 hours. And finally, the second isobutene measurement, then I unseal the vials and take samples for OD and for checking contaminations. Okay, so let's look at the result. So we can see that the isobutene was detected from so we can see that no isobutene was detected from all the KD minus strains, and there was isobutene detected from all both the KD plus strains. And as for the NS1 strain and the peak-2 strain, there was no significant difference between 0 and 5 hours. We can see that was less accumulation from the NS1 strain between 5 and 17 hours. Okay. So after this experiment, I noticed that there were some practical issues. So the first issue is that the measured peak area is really small. And the highest abundance I got from this experiment was around 300, compared this is far lower than the recommended amount. So to solve this problem in the next experiment, I decided to increase the starting OD, which in theory should increase the amount of isobutene produced, and also increase the volume of culture I put in the sealed vial, which will decrease the headspace volume, which in theory will increase the isobutene concentration in the headspace. Okay, and the other issue is that the method I use for measuring isobutene at 2 time points. So before the second isobutene measurement, I poked 2 holes, one in a DCMS measurement and one for taking the OD samples, which might cause some leaking and which might have made the accumulation in the second measurement to be underestimated. And the other issue is that in order to take the OD sample here, I have to stay in the lab at 9 p.m. using open flame, which is dangerous, was not recommended. Okay. So there's another issue that there was a really long pause that I take the culture of the shaker and then put them, measure the isobutene and then take samples. So this 6-hour pause, I just don't know how to factor that into data analysis and presentations. So basically, this is a really bad method to measure isobutene accumulation for multiple time points. So for the next experiment, I decided to just only measure the isobutene at 1 time point. Okay. So for experiment 3, so if you remember that I decided to put more cells into the vial. So in this experiment, I only had enough cells to make 1 sealed culture from 1 pre-culture. So the triplicate you're going to see are technical triplicates from 3 isobutene measurement in the same vial. Okay, so let's look at the results. So the isobutene concentration in the headspace was indeed much higher than the concentration in experiment 2. And if I compare the isobutene accumulation between the NS1 strain and the peak 2 strain, once again, NS1 strain showed much less accumulation, but I only have 1 sample, so it's not very representative. And now we can see that I detected isobutene from all the KTH minus strains, which is not supposed to happen, but I will leave the discussion for later. Okay. So after experiment 3, I noticed some experimental issues. So in these 2 experiments, I only used 1 transformance for each of the KTH plus strains. So it does not give us the complete picture for the performance of each strain. So in the next experiment, we use all the transformants I prepared. And next is the isobutene signal from the KTH minus control strain. So at that moment, I did not have the explanation or solution. So in experiment 4, I just tried to mimic the condition in experiment 2, where I did not detect any isobutene from the KTH minus strain. Okay, so for experiment 4, the procedure for each strain goes like this. And so I divided the cultures at the level of pre-culture to make it more representative of the biological variation. So let's look at the results again. So is there any difference among the three transformers of the same string? So it appears that there are some difference, but after running t-test and ANOVA, I found out the difference were not significantly different. Okay. And then let's compare the NS1 string with the PIC2 string again, and show that once again, NS1 string accumulated less isobutene than the PIC2 string. And finally, despite being a very small amount, I still detected isobutene from the KD plus strings. So finally, discussions. So the first thing to discuss the performance between genomic integration and the replicative vector. So from all the experiment, it is obvious that the isobutene accumulation from the NS1 integrated string is less than the PIC2 string. And this result was kind of expected based on the copy number of the chromosome in the synaptic system, which is actually lower than PIC2. And now the elephant in the room. So is that why was there isobutene signal from the KD minus strings? So the first logical explanation is cross-contamination, but the colony PCR result shows that the same. So this sample, I took them after the GC measurement, and the colony PCR result shows that the KD gene was not present for all the cultures of the KD minus strings. So which means that cross-contamination is very unlikely. Okay, so the next possible explanation is that the interference from other volatile compounds. So, yeah, okay, so since it was reported in the previous study that no isobutene was detected from the culture of Synechocystis without KD. So it's possible that some other similar volatile compounds were interfering with the signal of isobutene. So let's look at the chromatogram of KD minus string versus KD plus string. You can see that they have a very similar retention time. And if we look at the mass spectra, we can see that they have a very similar ratio between the 41 signal and the 56 signal. Okay, so for a compound to have a signal that's so similar to isobutene, it needs to have a similar retention index and a similar mass spectra where the 41 signal, the ratio between the 41 signal and 56 signal are similar. So the only compounds that fit these two criteria are the isomers of isobutene. So how do we separate the isomers when analyzing the volatile compounds in the headspace? Fortunately, the equipment GCMS I use in this study is capable to separate these. Okay, so in a previous study, the trans-2 butene and the cis-2 butene can already be separated from isobutene. But in that study, they did not manage to separate one butene from isobutene. Okay. So as we can see here, the retention time of isobutene in my study is less than the ones in the previous study. So which means that it is possible that at least one butene, the signal is interfering the isobutene, but my retention time is too low to have them separated. And if you dilate the retention time for even longer, you can separate one butene from isobutene. So basically it means that, yeah, you can separate the isomers using GCMs. You just need to run the GC for longer. Okay. And another hypothesis, this is a little bit far-fetched, that Synechocystis can produce isobutene natively. which is kind of unlikely because I have found no reports on cyanobacteria producing isobutene. Okay, but anyway, so for the context of this hypothesis, that in the mosquito study they confirmed that KICD can convert alpha-KIC into isobutene in vitro, or convert them into another metabolite HMB, and then HMB spontaneously decay into isobutene. And they also confirmed that, and was confirmed by me, that when you introduce RNKD into Synechocystis, Synechocystis can produce isobutene. Okay. And there are other enzymes that can convert HMB to isobutene in vitro. So now let's look at kind of far away with the degradation of tyrosine. So okay, so tyrosine is degraded into HPP, and in plants and mammal, HPP is converted into homogentisate by the enzyme HPPD, which is actually synonymous to KICD. And the mammalian HPPD and the plant HPPD has 29% similarity in the amino acid sequence. And in Synechocystis, homogentisate is a precursor for plastoquinone and tocopherols. Okay. And also there is a putative Synechocystis version of HPPD, but with a 34% similarity with the plant HPPD. But in this putative version of HPPD, it's only confirmed that it's responsible for the tocopherol biosynthesis. And in the mosquito study, it is confirmed that RNKD has a promiscuous activity in Synechocystis. They increase the concentration of homogentisate. Okay, so finally, let me get back to the hypothesis I want to propose. So if RNKD shows promiscuous HPV activity, it might be possible that the hypothesized Synechocystis HPPD has a promiscuous activity to convert alpha-KIC into HMB, and then may be possible that there are some enzyme or process that convert HMB to isobutene, which makes the native isobutene production route. Okay. But of course, this hypothesis is just very hypothesized, and we need to confirm that it is actually isobutene we detected from Synechocystis. Okay, so conclusion and future perspective. Okay, so in conclusion, that when RNKD is integrated into nucleoside 1, Synechocystis can produce isobutene, but it has a lower output than when the RNKD was expressed on peak 2. And then the KICD minus strains of Synechocystis can produce isobutene signals, and the isomers of butene could be responsible for this signal. And there are methods available to confirm the identity of this signal for future study. And so now for future study, it is the most priority to investigate the identity of this signal. And then once we confirm that it's not isobutene and it can be separated, we can investigate the performance of other KICD integrated strain that I have prepared in this study. And after that, we can introduce more copy of RNKD to increase isobutene production. And lastly, if it's confirmed that Synechocystis can produce isobutene natively, maybe we can investigate the possibility of the Synechocystis HPPD the possibility to produce isobutene natively. Okay, so yeah, that's my presentation. And I want to thank Karin Stanford for letting me work on this project to fulfill my dream to work on biofuels from cyanobacteria. And I would like to thank Barbara Bugal for her insight on scientific research and experimental design and how to write thesis and how to write presentation. I would like to thank Paul Hudson for being the KTH supervisor here. And I would like to thank all the members in the Cyano group at Uppsala University for providing me with the real lab experience. So yes, thank you for listening.